I would now like to introduce Emma Horrigan, Conservation Projects and Education Manager.
Hi, Noah, and hi, everyone. Thanks, Noah, for that introduction and the land acknowledgement. And thanks so much to everyone for joining us for the webinar this morning. As Noah mentioned, my name is Emma Horrigan, and I'm the Conservation Projects and Education Manager at Ontario Nature. And I hope after today's session, you will have a greater appreciation for the diversity of snakes found in Ontario and their unique ecology, and be inspired to get involved in this exciting long-term monitoring initiative. And with that, I am very pleased to introduce our speaker today, Brittney Vezina, who is Ontario Nature's Conservation Projects Coordinator. And Brittney has been involved with the Snake Long-Term Monitoring Project at Ontario Nature since 2019. She holds a Bachelor of Science in Zoology from the University of Guelph and a Master of Science in Conservation Biology from the University of Kent. And I hope you are as excited as I am to hear her presentation today. And with that, I will hand it over to Brittney.
Thanks, Emma, and thanks, Noah, for those introductions. And thanks to everyone for joining us today. It's great to see such a good turnout. So I'm just gonna get things started over here. Do I move that out of the way? There we go. All right. So yeah, as Emma said, I'm Brittney, and I'm the Conservation Projects Coordinator here at Ontario Nature, and I'm going to be walking you through the webinar today on Ontario snakes and our long-term monitoring protocol project. So before we get into the webinar, now that you know a bit about us, we would like to know more about our audience today. So we're going to run our first poll question, and please just select which group best represents you. So Noah, if you can run the first poll.
Is that working for you?
Yep. The poll is running.
Someone also mentions they can only see half the slide. I'm not sure if there's any adjustments to be made.
I'm not sure. I am in full screen and I swapped presenter view.
Someone else says the slide looks fine. So let me know when you'd like to end the polling.
Whenever it looks like the majority of people have answered, which it looks like we've got a pretty good amount now. All right, so it looks like we've got quite a bit of a mix in the audience, which is great to see. So just to give you a bit of an idea of what we're going to go over here. So we're going to start with just a quick intro to Ontario Nature and then some brief background information on snakes, their ecology, habitat, and threats. And then we'll go into the nitty-gritty of our long-term monitoring protocol and go over some snake identification. And then at the end of the webinar, we're going to open the floor to questions from the audience. So Ontario Nature is a charitable organization that has been running since 1931, and we aim to protect wild species and wild spaces through a variety of programs and projects that combine conservation, education, and engagement. And one such program is the Community Science Program, which has the long-term monitoring protocol project within it that has been running since 2017. Now, before I get into the details of the long-term monitoring protocol, it's important to highlight some background information on snakes, just to understand the importance of the project, but also to understand why the protocol is designed the way it is designed. So to start out, you'll see that Ontario has quite the high diversity of snakes, especially compared to the rest of Canada. We do have over 50% of the snake species in Ontario compared to the rest of the country. And over 50% of these species are at risk. And where they currently exist in Ontario is largely driven by environmental factors, namely being climate and temperature, though historical climatic factors and geological factors since the last ice age have also had a high impact on where you can find them today. But generally speaking, temperature and the number of snake species, temperature decreases as you go north and further from the Great Lakes, and also the number of snake species decreases. Of course, there are other influences that are specific to certain species depending on the type of vegetation or geology, but specifically speaking, generally speaking, the species decrease as you go north. And as I've alluded to, temperature plays an important role for snakes. So snakes are ectotherms, meaning they can only regulate their body temperature through behavior. So this means basking in the sun to increase their temperature, or moving out of the sun into shade to cool their bodies down. And so snakes, their behavior and activity throughout the year is largely driven by environmental conditions. So generally speaking, snakes hibernate in the winter months. They have to go below ground or somewhere where the temperature doesn't drop below freezing. And then as temperatures begin to increase in the spring, they will start to leave their hibernation sites and emerge from these areas, and with their behavior largely focused on basking during this time to increase their body temperature. And then as they begin to increase their body temperature, they will start to disperse across other habitats, where they will then reproduce and continue foraging and basking. And then with fall, once the temperatures start to drop again, they will begin to move back to their hibernation sites. So when it comes to habitat use, very generally speaking, snakes prefer open areas and edge habitats. And when I say edge habitats, I mean where two different habitat types meet. So this could be, as the picture shows here, forest meeting an open meadow. We've got an above ground photo and an on the ground photo to get a feel for this. And this is especially important in the early season when temperatures are low. These open areas give them a lot of room for basking in the sun to increase their temperatures, while also offering some cover from predators and access to prey while they're foraging. And within these edge habitats, there are a lot of microhabitats that the snakes use, specifically natural and artificial cover objects, which are just objects that they can take shelter under. They are important throughout the active season. They use them for resting, foraging, shedding, hiding from predators, but also with thermoregulation, as these objects can absorb heat and retain heat. So this can include down woody debris, rocks, tin, or things like plywood boards. And so snakes in Ontario are unfortunately, and they face many threats, including habitat loss and fragmentation and road mortality, which is specifically quite high in Southern Ontario, where there's a lot of people and a lot of roads, as well as persecution due to fear or misunderstandings, poaching for the pet trade, and disease. But snakes do play a very important role in ecosystems as predators of pests. And so there is a high need for conservation action for these animals. But unfortunately, snakes are very cryptic animals. They like to hide, and they can be difficult to find and study. So because of that, there's a lot of information lacking on population trends for many species. And this makes it difficult for us to assess their conservation status, especially for the Committee on the Status of Endangered Wildlife in Canada, as they need population trend data to effectively assess a species. So what can be done in this instant for snakes? Now, it is recognized that there is a need for standardized, widespread, long-term monitoring to help gather this baseline information and fill these knowledge gaps. And despite being cryptic species, there are methods that exist that can help detect them. And so for these reasons, Ontario Nature developed a long-term monitoring protocol for assessing trends in Ontario snakes over time. And we did this with in partnership with experts, conservation professionals, and landowners across Ontario since 2017. Now, specifically, our monitoring protocol uses cover board transects for collecting our data, or as I've mentioned, they're also called artificial cover objects. So these objects are simply placed out in habitat for the purpose of sampling animals that take cover or shelter beneath them. And for this protocol, we use cover boards that are set up consecutively in a line, or what is called a transect, with each transect of boards then representing a random sample or a random snapshot of snake species that are present in that habitat at that point in time. So in this case, this involves 24 4-foot by 2-foot plywood boards that are spaced 5 meters apart. And ideally, based on the ecology of snakes and their use of open habitats for basking, you want them to be set up in full sun. And ideally, you also want them to be set up in the fall prior to monitoring in the spring to allow the boards to weather, as snakes prefer weathered boards. But at the very least, 2 weeks prior to monitoring is ideal. And so why do we use cover boards? This is something I've already gone over a bit, but there are multiple benefits to using cover objects set up in this way. So one of which is very important, is making sure that our monitoring efforts are standardized across sites in Ontario. So this means that surveyors are following the same methods and taking the same random sample or snapshot over the same time frame at the different sites. And this helps to reduce observer bias, and it makes it more possible for us to draw conclusions about any trends we may find in the future across sites, but also across time. And cover boards also help to reduce habitat destruction relative to if you're flipping natural cover objects like woody debris in a habitat randomly. And of course, cover boards are also incredibly important to the purpose of our research, as they do increase the chances that you're going to find snakes, because they, as I said, they use natural cover objects for many reasons, including to thermoregulate. So if you're strategic about where you place the cover boards and when you check them, you're very likely going to find snakes under them. And of course, I should note that cover boards do have a relatively high initial setup cost, especially these days with the cost of lumber. And they can take significant effort to set up, but once they're out there, once they're purchased, they can last many years and they require very little upkeep. All right, so we're gonna run our next poll just based on the information that we've talked about so far. I would like to know what you think is the most important step in site selection. So when you go out and you select where you want to put your transect. So we've got the poll open now. All right, it looks like we've got the majority of people answering now. And the majority of you said all of the above out of habitat, sun exposure, recent snake sightings, and accessibility. And that certainly is the best answer, as they're all very important things to consider. But this is one of the most important steps out of this protocol, is selecting your site, as obviously transects need to be in appropriate habitat to increase the chances of detecting snakes and in turn collecting enough data that we can detect trends over the long term. And some of the best advice I've been given when working on this project is to think like a snake. You want to look at your habitat and think, where are snakes going to exist? And so this is most often in open, as I said, open or semi-open areas with ground vegetation and in a spot that gets sun exposure during most of the day. And as I said, snakes prefer edge habitat, especially where field, marsh, or rock barrens meet. And suitable vegetation communities typically used by snakes include forest clearings or edge habitat, open canopy swamp, marsh, alvar, rocky outcrop, dune, savanna, and meadow. And of course, if you've already seen snakes in the area, that's a great indicator of good habitat. And you do also need to consider accessibility in terms of, do you need specific permission or permits to do research in that area? Are you able to access it regularly to monitor? But also, what is the access to the general public like? If it's a very high traffic area where the boards will be very visible, it's not the most ideal location, as poaching is a major threat for snakes. And one tip that I can offer is Google Maps or Google Earth is your friend when you are going through this process. It really helps give you a bird's eye view of the habitats in your area where you can identify those open areas. And I should also note that the process of good site selection may involve some trial and error as you start monitoring. And so the next most important step is determining when to begin your monitoring season and when during the season to conduct your surveys. So our ultimate goal is to maximize the number of snakes that you're going to come across. But in reality, there are a number of factors in nature that are going to influence whether or not this happens, from population density to behavior, as we've mentioned, and observer experience. So to help reduce this bias, while also maximizing the probability of capturing snakes, we set strict weather and timing guidelines in which to conduct surveys. And this goes back to standardizing data collection and aiming for consistency across our sites. So again, you want to think like a snake. Ideally, you want to begin monitoring once snakes have started to disperse from hibernation. And they will start doing this in the early spring once overnight temperatures are above freezing and once daytime temperatures are sitting at around 10 degrees. And so this will depend on where you're located in Ontario. Obviously, further north, you're probably going to be starting monitoring later, but the dates we offer are merely just guidelines. We recommend, touching base with naturalists in your area and, Facebook groups like the Reptile and Amphibian Atlas Facebook group to get a feel for when snakes are on the move in your area. And additionally, the protocol calls for 8 surveys over 76 days, so roughly every 5 to 9 days. And this is to capture snakes before the temperatures increase and snakes become more dispersed in July and August, and also less likely to be found under cover. And so we also have strict guidelines in terms of the weather conditions and the time of day that we conduct surveys. So in terms of weather and air temperature, you want to aim for between 10 and 28 degrees, as if it's too cold, snakes may go back underground or may not be on the move. But if it's too hot, they may not use cover boards, or they also may go underground. And as well as not too high of winds, but wind isn't as much of a consideration that is concerning, and no rain. And time of day is within 2 hours after sunrise or 2 hours before sunset. And this is to try to capture the snakes while they're hunkered down or while they start to hunker down for the night before they are on the move again. And sometimes it is tricky to have the weather and the timing window align, especially early in the season. So if there's ever a conflict, we always recommend to stick with the time of day, so that 2-hour window. And so, of course, the next step is going out and collecting your data. So there's a number of variables we have participants collect, including multiple weather variables at the beginning and end of survey, as well as total survey effort. So we ask individuals to use stopwatches to record their effort. And This is because in the end, the data is calculated by unit effort. So it's standardized by survey effort, as increased searching time typically leads to more snakes. And then once you reach a cover board, you'll lift the cover board, kind of look visually, but also move your hands throughout the vegetation. As you can see here, snakes can really get hidden in that vegetation, and it's important that you try to capture all of the snakes that you find under there. And when you find snakes, of course, you want to identify the species and the number of individuals. And then if you run into a species at risk, we ask for photos and aging and sexing the snake. And if you are unable to age or sex the snake, this is where photos, especially with a size reference and the underside of the snake showing the tail and vent, is important. And so one of the challenging aspects of this project can be identifying snakes, depending on your experience, but it's more so because sometimes snakes can get away or you only get a quick look at them. So I will be going over some of the species that are most likely to be found under cover boards, but also that are most likely to be confused. So one of the most common snakes you're definitely going to find under cover boards is the eastern garter snake. It's a very widespread snake that is common in many places, but it can sometimes be mixed up with the Butler's garter snake or the eastern ribbon snake, which are both species at risk. As all three of these snakes have the yellow to cream stripes, one along the back and two along the sides, and a generally dark background. So the most important things you want to look for are your location. Where are you in Ontario? Eastern garter snakes are very widespread, whereas Butler's garter snakes are very restricted in their range and are only found in isolated pockets of southwestern Ontario. Eastern ribbon snakes are a bit more restricted than eastern garter snakes, but they're specifically related to wet habitats. And next step, you'd want to look at their head. You'll see that the Butler's garter snake has a smaller head than the eastern garter snake, and the eastern ribbon snake has a white crescent in front of the eye. And while it isn't as obvious, you can also confirm the species by looking at the scale row where that lateral side stripe falls. So it is a very subtle difference. So this is where pictures really help, or having the snake in your hand. But the eastern garter snake's stripe is the lowest, closest to the belly. The Butler's garter snake stripe is a bit higher, and then the eastern ribbon snake is the highest on scale rows 3 and 4. And so if you're ever unsure, especially since we have species at risk involved, we recommend that you take pictures of the side of the snake like this. And you will likely also come across some of Ontario's smaller snakes, including the Decay's brown snake. So it can reach 20 to 50 centimeters in length and is generally gray, beige to brown, with a beige underbelly, though sometimes the underbelly can be a bit of a tinge of pink, at least what I've found out in the field. But the distinguishing features you want to look for are 2 rows of spots, or some people say look like diamonds, running along the back of the snake, as well as small dark bars behind the head and a blotch beneath the eye. And then as neonates or newborns, Decay's brown snakes look very similar to adults, but they have a white ring around the neck that will fade with age. And another small snake, which could be mixed up with Decay's, is a northern red-bellied snake. And so the northern red-bellied snake has a very distinctive red belly, but there's definitely instances where you may not get your hand on that snake. So you may not see that red belly. And so if that happens, you want to be looking at the back of the snake. If you see 4 thin dark stripes, at least the 2 on the back, there's also 2 along the sides, but those 2 stripes along the top will help you distinguish from Decay's, whereas the Decay's has the spots or the diamonds. And another feature is the diamond-shaped blotch on the neck of the red-bellied snake, which can be found both on adults and neonates or newborns. And the next general group that is probably the most difficult to differentiate are the blotchy snakes, which are also typically the largest snakes in Ontario, and also ones, this general group has a lot of species at risk. So it's really important to learn about the different ones you might run into and how to differentiate them. So the eastern fox snake is one of these snakes. We've got an adult on the left and a neonate or newborn on the right. So with the adult, they are one of the largest snakes in Ontario. They can get up to 180 centimeters in length. And you'll see adults have a very distinct copper brown head. They also have a yellow to brown background with large dark blotches running along the back and along the sides. And eastern fox snakes have keeled scales. So snakes can have keeled scales or smooth scales. Keeled scales will have a bit more of a rough appearance to them due to a ridge that runs along the middle of the scale. And you will also feel it if you have the snake in your hand. It is a much rougher feel, not very rough, but there is a difference between a keeled scale and a smooth scale in your hand, but also visually. And the neonate or the juvenile eastern fox snake, you'll see will lack that copper brown head, though as juveniles, they may start to form that coloration. And you'll see that the blotches are brown, but have a bit of a dark border, but they will still have the keeled scales. And another blotchy species at risk that could be confused for others, including the eastern fox snake, is the milk snake. So the milk snake does not get quite as large as the fox snake. It reaches maximum about 130 centimeters in length, but it can look similar. So it has a gray to light brown background, also has large blotches running down the back and the side, but these blotches are more red or brown in color and have a dark border around them. And milk snakes will have smooth scales, so they will feel smoother and look smoother, but you won't see that ridge through the middle of the scale. And not all milk snakes will have this, but some will have this Y-shaped blotch on their head. And when it comes to neonates or juveniles, as pictured on the right, they are very similar, though the blotches will be more reddish in color. So when distinguishing between the fox snake, it really comes down to the color of those blotches, the smooth scales, and generally speaking as well, milk snakes are more slender bodied than fox snakes that are a bit thicker bodied. And a snake that many are familiar with in Ontario is the Massasauga. And it's a species that many may confuse other blotch species for, including the milk snake. This is understandable, as you'll see, the Massasauga has a gray to brown background color and also large, dark bordered brown blotches running along the back. But you'll notice that the blotches are a very distinct saddle shape. And the Massasauga, as you'll see, also looks very rough. They have very keeled scales and is very thick bodied, especially compared to the milk snake, but even more thick bodied than the fox snake. And of course, rattlesnakes also have their distinctive rattle, though this sometimes falls off, so you can't always rely on this. And as a viper, they have vertical pupils and the heat sensory pits and stripe markings that run alongside their wide head. And the neonate juvenile is very similar to the adult, but will have fewer segments on the tail. And people may say the vibration of the rattle is distinct, which it is certainly distinct, though you should know milk snakes and fox snakes may also vibrate their tail when they are threatened. And the final snake that I will go over is another blotchy snake and another species at risk, the eastern hognose snake. Now, the eastern hognose snake has colors and patterns that are highly variable. Some adults have solid colors from gray, green, orange, yellow, whereas others have large dark blotches running along their backs. But one thing that they always have is the dark blotches behind both sides of their head, and always the distinctive upturned snout. So this is something to look for. They also have keeled scales and are a bit thicker bodied. But again, looking for that upturned snout is important, and also behavior is huge for hognose, as they will typically hiss at you and potentially also play dead. And the bottom there is a picture of a neonate. And again, they look very similar with the black blotches and the upturned snout. So now we're going to run a few polls to test your knowledge and see if you are able to identify some pictures of snakes. So here we have a picture of a snake. If everyone can let me know if they think it's a DeKay's brown snake, a ringneck snake, a red-bellied snake, or none of the above. Okay, looks like the majority have made their decisions. And it looks like the majority thinks this is a DeKay's brown snake. And you are correct. This here is a neonate DeKay's brown snake. So you can see it has, though it is a bit hard from this angle, 2 rows of dots or diamond shapes along the back. And it also has that black marking behind its eye and below its eye. And as I mentioned, the neonate DeKay's brown snakes have a white ring around their neck. And if you had this guy in your hand, you could also flip him over to see, okay, he doesn't have a red belly. So good job on the ID. We will move on to the next one. What species is pictured? We have a gray rat snake, a milk snake, a Massasauga, excuse me, an eastern fox snake, or an eastern hognose snake. Looks like people are much more quick to decide on this one. And the majority of you said an eastern hognose snake, and you are correct. This is an eastern hognose snake. A bit hard to tell from this angle. I did try to make these a bit tricky, but it does have the upturned snout and the 2 black blotches past its face there. And if you were there with me in the field that day, you would have heard a lot of hissing, so you would have known for sure that it was a hognose you're dealing with. And we will move on to the next one. What species is pictured here? So do we have a gray rat snake, a milk snake, a Massasauga, an eastern fox snake, or an eastern hognose snake? And it looks like the majority of you chose milk snake, and you are correct. This is a neonate or newborn milk snake. And you can tell because of those reddish blotches with the dark border. As well, it's a bit hard to tell in the angle of the photo, but it did have a Y marking on its head. And as well, some people said eastern fox snake, but this guy was quite smooth and had smooth scales, and it's pretty slender. But it's really those blotches, the coloration of the blotches, and that Y shape that you'll be looking for. Okay, good job, everyone. And we'll just pivot back to the protocol. Now that we've gone over snake identification, our protocol also includes data collection on habitat of your transect site. So methods involve classifying the vegetation communities that are dominant at your site and to track any changes that occur to the habitat over time, as well as any disturbances. So it's very high level ELC, including dominant canopy type, age class of any trees or shrubs, and the overall community class, as well as just a general description of any disturbances and vegetation in the area. And of course, we cannot collect data without the fun of statistical analyses. So we plan to use count data, so number of individuals, as an index of abundance to produce a relative measure of population size based on survey effort. And this is to help us track trends across snake populations over time. So the data will be analyzed using an open end mixture model developed for estimating population size from count data. And we have found so far with the project since 2017 that our cover board setup and our protocol can capture a large number of snakes if the transects are placed in good habitat. So what we are aiming for now is further years of data collection to add to this long-term database at our current sites across Ontario, and also to add more sites across more habitat types. Our hope is to continue building partnerships with individuals and organizations in implementing this protocol over a larger scale basis with the aim of detecting population trends in snakes in Ontario over a 10-year period. And this will help us to fill knowledge gaps and help Coast Seawick in assessing the conservation status of both species at risk, but also common species. While I've outlined the data we generally collect using the long-term monitoring protocol, there are other ways that the protocol could be beneficial to your work or your general interests, whether it be a site that you manage or just your own personal property. So generally speaking, the cover boards can help you gain presence data, excuse me, and get a better understanding of species that occur at a site. It can also be applied to monitor changes in abundance of snake species before and after habitat restoration, such as invasive species removal or prescribed burn. And you can also use marker capture methods to monitor population demographics. So in terms of adults, juveniles, neonates, but also males, females, and their survivorship over time. But this does require marking individuals, which is possible using things such as pit tags, though this does require special training and permits. But you can also mark and monitor individuals in non-invasive ways, such as monitoring individuals by using unique blotches for certain species, such as the one pictured here is an eastern fox snake, fox snakes, milk snakes, and Massasauga rattlesnakes. They have unique blotches. Each individual has unique blotches, and especially around the head. So this is something that you can monitor over time to see if you are recapturing the same individual year after year. And as I said, we are looking for more individuals and organizations to apply this protocol in a variety of habitats across the province. And there is a general level of skills useful for participating, including a general understanding of snake biology and ecology, which we have briefly gone over during this presentation, as well experience generally handling snakes, aging and sexing them, and identifying species is obviously a great asset, as well as plant identification skills. But whether or not you currently possess these skills, it doesn't hurt to reach out to us if you're interested in participating. Specifically, we are tentatively planning in-person training opportunities in 2 regions of Southern Ontario, one at one of our sites in Norfolk County, geared towards training for people in and around Simcoe, and one at our Sydenham River Nature Reserve, geared for training opportunities for people in and around Strathroy. And we're aiming for late June, early July, or fall. It really depends on what happens with COVID-19 restrictions, but it will include hands-on training on our protocol at one of our transect sites. We'll run through the protocol, and obviously if we capture snakes, we'll be going through snake sexing and identification, and also some information on transect placement and habitat data collection. So if you are interested in this training opportunity, please do reach out to me at brittneyv@ontarionature.org for more info. Even if you are interested in participating, but you're unsure if your region qualifies or if you're able to attend in-person training, please don't hesitate to reach out. There may be other ways that we can support you in gaining the skills that are helpful for training. And so on that note, before wrapping up the webinar, we will run the final poll to see how many of you out there may be interested in joining our efforts. Good to see. We have quite a few of you responding.
All right. Yeah, there have been lots of great questions so far. So we can just go through some of the questions that folks have had.
I was just going to quickly run through just the thanks. I know we're running a bit low on time.
Yeah, sure. Go for it.
Before we do get into questions, yeah, it's great to see a lot of people interested. So we will get into questions shortly. But yeah, I cannot end the webinar without thanking those who made the project possible. We offer many thanks to the conservation professionals and experts who have provided guidance and feedback throughout the pilot stages, the development stages, and to the many individuals and organizations who tested the protocol and continue to apply it today. And we also owe many thanks to the many organizations who have provided financial support for this project over its lifetime. And we offer a special thank you to TD Friends of the Environment Foundation for generously supporting this webinar and the related training events. And yeah, of course, I can't go without thanking everyone who's in attendance as well. We are going to open the floor to questions now, but if there isn't a time to answer all of them, you can definitely reach out to me afterwards and we can discuss further. Thanks, everyone.
Okay, thank you so much, Brittney. That was lots of great information, and lots of folks who joined us this morning have a number of questions about different aspects of the presentation. So one question that came up was around minimum transect length. So an individual was just wondering there, is there a minimum length that the transect needs to be?
That would include some quick math on my end. The protocol is designed using 24 cover boards. Ideally, with the way it was designed in terms of statistics, it is supposed to use 24 cover boards, and they do need to be 5 meters apart from edge to edge. So there is a minimum length, if I can quickly do some math, but we have run into issues with some partners saying that they don't have the length for a linear transect with that many cover boards. And this is something that we are working on discussing on how we can modify the protocol to work with different areas, different amounts of space for the cover boards. So yeah, if you think you don't have the space, still please reach out and we can discuss more detail in terms of what you may be able to do instead. And I can probably give you a better assessment of whether or not you have space, because I'm not good at quick math.
Yeah, I mean, if I think having people who don't think they would have the space necessary to set up a linear transect, if that's the situation you're in, just contact us and we can kind of work through what the various options are. So then Lance is wondering about painting plywood on the exposed side, either white or black, and if you set up a transect, if that's something that would be beneficial to do.
Yes, so this is something we do and we recommend for our partners to do as well, especially, to mark the numbers so you can keep track of the numbers as you're monitoring, but also just to let people know from the general public, like, please do not disturb these cover boards. So we use bright orange paint on the exposed side, and yeah, we use like a stencil to spray paint for research, and the numbers 1 to 24.
Okay. I think too, also that question was getting at, in some cases, people will paint cover boards black to help with heat retention.
Right.
So painting some kind of marker on them to identify that they are part of a research project is a great idea. In our case with the protocol, in terms of painting the boards black, that's not something that we've done, although there are certainly many projects that do paint boards black, but just the logistics around and the cost associated with painting the boards is what led us to not incorporate it into the protocol at the end of the day, but it certainly is an option that people can use. So this next question is from Janet. If you had several snakes cohabiting under one board, would you recommend having a pillowcase handy to capture them for the short time it takes to process info and collect all the relevant information, including photographs?
Yes, definitely. We typically use pillowcases. You could also use like plastic bins, but it's harder to keep the snakes in there, whereas pillowcases, you can just throw them in there and tie it shut and then go species or individual snake by individual snake. But yes, at some of our sites that we personally monitor, you can come across many snakes under one board, upwards of about 12 to 15. And especially once the temperatures are getting warmer in the June months, it can be really tricky to grab them all and to make sure that you are recording every single one that you see. So we definitely recommend bringing pillowcases with you. The only thing is, you want to make sure you aren't using the same pillowcase between sites, and you want to wash them between visits.
Great. And sort of tying into that question, Toby is wondering if you could comment on the fact that we're asking people to handle snakes. Can you go over the ethics of handling them and how to handle them properly?
Yeah, and there are details in our protocol regarding this for more information, and definitely something that anyone involved in the project should review ahead of doing any monitoring. But yes, typically with snakes, you want to allow them to move within your hands. So kind of holding them closer to the head and further down along the body so that it doesn't stress them out too much. They feel like they're still on the move. And yeah, minimal handling, just enough handling to get the data that we need for the project is ideal. And then you want to put the cover board down with no snakes under it because coverboards can squish them if you put them under the coverboard on the ground and then put the coverboard back. So you want to put the coverboard down and then allow the snakes to move back under the coverboard on their own. But of course, before even handling snakes, you should also be following decontamination protocols in terms of sanitizing your hands, making sure there's no bug spray, no sunscreen, that sort of thing, as they can be very sensitive, and also disease transfer is a major issue with snakes. So just making sure that yeah, you're sanitized beforehand is important. And again, there's a lot of detail about this information with links to relevant resources within our full protocol. So, and on that note, many of the things I've gone over are provided in much more detail within our protocol. And this is something we can share with interested individuals afterwards.
Great, thanks. So David is wondering, because the individual sizes of different species of snakes varies, does coverboard size have any effect on survey results?
Yes, so that is a good point. And I know this was something that was looked into a lot before deciding on our specific protocol in terms of what size of board to use. Because yes, the larger the board is, the more ideal for larger snakes such as fox snakes, rat snakes, etc. And in the end, it was decided to go with our size board as it is big enough they found in the literature it is big enough to capture small snakes, but also in conversations with individuals doing research in Ontario, it has also been big enough to capture snakes as big as fox snakes, which was the original species that was being targeted for this protocol. And we have been successful at capturing fox snakes under our boards, but yes, there's certainly going to be some limitations to the protocol. We aren't necessarily going to be able to capture every species, but yeah, as I said, it was initially designed targeting fox snake, which is the second largest species in Ontario.
Great. And one individual is asking which snakes that you showed in your presentation are venomous.
Good question. Yes, just the Massasauga. The Massasauga is the only one that is relevant to humans in terms of being an issue in terms of that, sorry. And actually related to that, if you are monitoring in an area where there may be Massasaugas, and our protocol goes over this, you are supposed to lift the cover boards away from you so that you are protected if there's Massies under there. And there's also information on if you were to be handling them, how you would do so using a snake hook.
Okay, and there were a couple of questions around the listing of milk snake. And so I wondered if you could just comment on the current status of eastern milk snake.
Sure, yeah, so they aren't listed provincially, but they're listed federally as special concern, I believe. Not sure if you can add to that, Emma, but I'm pretty sure that's their current listing. So we would want information on milk snake from the monitoring program due to this.
Yeah, that's correct. So it's still currently listed federally as special concern, and we treat it like all the other species at risk. So we collect photographic data, and in the protocol, it's outlined as a species at risk. Okay, another question from Rob about whether there are any geographical areas where data is lacking thus far. For example, Sault Ste. Marie, Espanola, sort of Northern Ontario. And do we have a general map showing where these gaps or holes in data exist?
Yeah, that's a great question. We don't currently have monitoring further north than, thinking Ontario geography, along the lines of like Georgian Bay, I believe. So yeah, we are certainly lacking monitoring in the north. And we don't currently have any publicly available map, but that would be a great thing to add to our protocol, is the distribution of our current monitoring sites, so that people could get a feel for where we may need more monitoring. That is a good idea.
Yeah, and I guess if you follow up with us directly, we can help give you a sense of where our current transects exist and where we'd be looking to fill in those gaps. So we can kind of go back and forth with you there. I'm just looking at the time, it's 12 o'clock. Maybe we just have time for, just looking here for one more question. So will the protocol be provided in full as a PDF for participants? And you kind of alluded this in some of the earlier questions, that if folks are interested in getting involved and participating in this project, to contact Brittney directly at the email shown just on the slide. And so Brittney v@ontarionature.org, and she'll be able to follow up with the full protocol for you to read through and answer any questions that you have. And yeah, that pretty much concludes our time for the Q&A. Thanks so much to everyone for your questions and joining us for the webinar. Sorry we weren't able to get to all of them, and I'll hand it off to Noah for closing remarks.
Hello again. As a reminder, in the coming days, in case you would like to rewatch this webinar or share it with your friends or colleagues, the webinar video will also be available on our YouTube channel, and we will send a follow-up email to everyone who registered for this webinar. Many thanks, especially to Brittney for this LTMP presentation, and to Emma as today's host. And thank you all for attending and learning about long-term snake monitoring protocol. Have a great day, everyone.
Thanks. Bye.